We would like to show our respects and acknowledge the Bedegal people who are the traditional custodians of the land on which this meeting takes place, and to elders past and present.
Single cell PIP6 and Parse statistic and scale bio quantum scale, but today we are excited to focus on Parse statistic application. Just to give you a summary of what's happening, we have the showcase presentation by Dr. John Thompson, who walk us through the Parse statistic technology, and then we have Dr. Osvaldo Contreras, who will present his research using the Parse statistic, and then we have also afternoon tea, so please stay along for further discussions and chats and questions. And the afternoon tea is provided by the team. So it's going to be a very exciting event, and I'd also like to introduce John. John is a Parse Biosciences field application scientist, and he has earned his PhD at Duke University studying liver regeneration and completed a postdoc at UC Mercer, focusing on single-cell genetics and epigenetics. At Parse, John supports scientists with experimental design, sample prep, single-cell workflows, and data analysis. So, please, John.
Thank you for that lovely introduction. So yes, I'm John Thompson. I'm a field application scientist from Parse Biosciences, which means I help people run single-cell experiments. So today, let's talk about Parse single cell. Now, for today's agenda, we'll do an overview of the Parse Evercode technology. We'll do a comparison of Parse split pool combinatorial barcoding versus droplet-based single cell. We'll talk a little bit about Evercode immune profiling with TCR and BCR. And then we'll do some conclusions, and after that, I'll pass it on to our guest speaker for today. Now, because this is a slightly larger crowd, let's go ahead and hold questions until the end. So if you have any questions, please write them down, and we'll address them near the end of the presentation if we have time. So, Parse is in over 2,800 labs currently and growing. And actually, this number is probably closer to 3,000, not quite 3,000 yet, but we're getting close. So one amazing thing about Parse split pool combinatorial barcoding technology is the scalability. Using split pool combinatorial barcoding, we can scale up infinitely, and this is just one experiment that I was part of. We were able to go from seeding cells to data back on over 100 million cells in 3 weeks. So this is a major atlas project run by Tahoe Therapeutics with over 50 different cancer models multiplexed together, over 1,100 drug treatments, over 56,000 individual experiments and drug treatments, culminating in this massive 100 million cell data set to train AI models on how cancers react to different drug treatments and combinatorial drug treatments. This was a really fun project, and if anybody has any additional questions, feel free to stick around and chat during the tea. What's amazing about this, though, is if you were to run this type of experiment on another technology, you'd get so many batch effects that you can't really compare all the samples together. Now, they ran the batch effect analysis, and this shows the batch effects. Just minimal batch effects between each of the samples across all of these different samples, 100 million cells. So the ability to scale up exponentially can really power these cancer studies, can power these AI studies, and can really help with discovery, especially to help with learning more about human health and treatments for specifically cancer research. Plenty of other applications, but this is one of my favorites because I helped out with this. So, introducing Evercode technology. I brought up the scalability. It's the same workflow, whether you're doing a small pilot experiment for 10,000 cells or a larger small atlas experiment with 5 million cells. And you can easily, the workflow is pretty much the same, whether you have one sample or you have 384 samples. The hard part there is the sample prep, depending on how many you got. Unmatched data quality. We see higher gene and transcript sensitivity, and you avoid doublets and ambient RNA just from the inherent technology. And I'll go over that in a little bit of detail in a bit. No instrument required, so you don't get any clogs, you aren't limited by your cell size, you're not going to lose your entire sample if a little fluidic channel it gets blocked. There are also flexible automation options if you want to have a robot do the assay for you. And there's no bicycle pump with an iPad screen running only one proprietary technology. It's powered by fixation, which means you get your samples, you prepare them, you fix them, and then you can store them until they're all ready. And you can lock in the biology for up to 6 months while you're preparing samples, then barcode them all at the same time, whether that's in your lab or with the local core. So the main portions of the Parse technology is fixation. So you have your fixation kits. One fixation kit fixes 12 samples, and you fix the samples on your schedule. You'll receive the samples. Sometimes you receive them at an expected time. Maybe you're using mouse studies and you are able to dissect your mice and collect your samples and you know when you're going to get them. Sometimes you're doing human studies and you get the samples when you get the samples. It makes it easy to prepare the samples when you get them, fix them, bank them, and then transfer them over when you're ready. So no rushing to the core at 10 p.m. because you just got those samples and they have to run it now. Then the barcoding and library preparation kits will allow a unique barcode to be added to each of the transcripts in each individual cell, and I'll talk about that mechanism in a little bit. Sequencing, so Parse technology is sequencing agnostic, whether you're doing short read sequencing via Illumina, MGI, Ultima, Element, Singular, etc., or long read sequencing, such as with PacBio or ONT. And we also have our Trailmaker data analysis platform, which allows biologists to analyze the data, to do the clustering, to generate your figures, go all the way from FASTQ files to publication-ready figures without knowing any lines of code. Now, if you've used any type of these platforms, you know that these can be kind of clunky. Thankfully, Trailmaker was made by teams who tried some of these other user interface platforms, realized that it's clunky, unusable, decided they wanted to make their own, and this actually is very easy to use. So, Evercode technology, the way you want to think about this is the kits come in different sizes. Mini kit for 10,000 cells, WT kit, a whole transcriptome kit for 100,000 cells, mega kit for up to 1 million cells, penta kit for up to 5 million cells. And you're going to split that across up to however many samples it says here. So for example, a mega kit, 1 million cells, you can split it to up to 396 samples, or you can split it up with 384 samples. So if you do a 1 million cell kit, split it in 96 samples, and you get 10,000 cells per sample. The basis of the technology is the whole transcriptome, but you can also add TCR, BCR, crop-seq CRISPR screens, or gene pull-downs, and I'll talk about these two specifically in a little bit. So, the way Parse technology works is that we're using the cell itself as the reaction vessel. So here, we have our single cell suspension, or a single nuclei suspension. We're going to fix them to lock in the biology, and then we're going to permeabilize them, which allows the biochemical reactions to occur inside the cells themselves. So the enzymes go into the cells, the barcodes go into the cells, and the cell itself is the reaction vessel, which would be critical for the technology, which I'll talk about in a bit. But the fixation itself takes about an hour per batch. A batch can be 1 sample, a batch can be 96 samples, and anything in between. Whenever you have your samples ready, that's when you're going to run the fixation. Then you can store them in the -80 for up to 6 months. Once you have all your samples ready, then you can easily transport them to the core on dry ice. And then the core can run them when they are ready to run all of the samples. So it eases a lot of the logistics of sample collection, of running your single cell experiment, of all these time course studies or collecting the samples at different times. Fixation is the answer to that. So the way the combinatorial barcoding works, and this is key to the technology, you have your fixed and permeabilized cells here. And this example is our mega kit, our 1 million cell kit. So for our mega kit, we have 96 wells in our first 96 well plate. We're going to load our samples into a well of the 96 well plate, and we're going to perform a reverse transcription reaction that occurs inside the cell. So we're going to reverse transcribe the mRNA using both an oligo DT primer to get 3' information and random hexamers. And these hexamers allow you to get information throughout the transcript, not just on the 3' end. So you get SNPs, you get splice variants, you get more information as opposed to just 3' oligo DT information. Also, attached to this reverse transcription primer is this well-specific barcode number 1. This well-specific barcode number 1 will tell us which well a specific cell was loaded into. So if we know which sample we loaded into the well, and we know the sequence of this well-specific primer, therefore this first barcode is a sample-specific barcode that we'll use to demultiplex later on. The next step is going to be pooling everything together from all the wells, all the samples. That multiplexes everything together and will minimize the batch effects. And remember, we're going to demultiplex via this first barcode later on. Next, we're going to split into the second barcoding plate randomly, ligate on a second barcode, pool, split, ligate on a third barcode, pool, give them a wash, count them, split them, and then load them into what we call sublibraries. Then we'll add a fourth barcode when we're in the sublibraries. So in this way, the first barcode is a sample-specific barcode. Second and third barcodes add complexity and diversity. And the fourth barcode is a sublibrary-specific barcode. And what's great about these sublibraries is you can sequence one of these sublibraries first, take a look at the quality, maybe oversequence it to look at your sequencing saturation, figure out how much you need to sequence before tossing in a lot of money to sequencing all of these sublibraries if you want to sequence them deeply. You get an idea of how your experiment's going to look before you jump all into the sequencing. What's also really nice is the number of cells you load into the sublibrary is how many cells you're going to get. So you can accurately determine how many reads you want to throw at each sublibrary, not just throw a handful into a machine and hope you get a certain amount back. You will know exactly how many cells you will get from your data from when you're loading your sublibraries. And with this, the number of possible barcodes is 96 times 96 times 96 times 16. Gives us over 14 million possible barcodes, but we're just going to use 1 million. And that allows us to minimize the doublet rates. So what that means is even at the 5 million cell experiment size, our doublet rates are less than 2% because of the combinatorial barcoding. Now, nowadays, single-cell RNA-seq studies are getting larger and larger and larger. And this is needed to identify rare cell populations and increase your statistical power for your downstream analysis. Because for a single-cell experiment, n equals 1 is 1 cell, and the more cells you have, the more statistical power you have, and the more convincing your data is. So this is just a standard Parse experiment. It's a million cells, our standard mega kit, 12 samples, and this was completed in 2 days for the wet lab portion. We took 12 PBMC samples. We fixed all 12 samples simultaneously using plate-based fixation, and then performed EverCode barcoding in 2 days. The workflow can be done in 2 or 3 days, depending on how you want to set it out. For a first-time user, I usually recommend doing it in 3 5-hour days, but an experienced user such as the core can possibly do turnaround in 2 days. Now, I can't promise anything, of course. It'll depend on the queue. Now, then after generating the single-cell libraries, they sequenced it on NovaSeq X and then ran the bioinformatic analysis on our easy-to-use TrailMaker platform. What we saw is, and this is a UMAP representing over 1 million cells. And you'll notice that there are some of these clusters that are barely any cells here. Even though this is a million-cell experiment, you don't really have too many of these. But let's talk about that a little bit. So of course, being a PBMC data set, you see your classic immune cells, your T cells, your B cells, your monocytes, etc. But let's zoom in on this top 80% or so, these rare cell populations. So from this data set, since we have 1 million cells divided by 12 samples, that means we'll get about 80,000 cells per sample. So for some of these rare cell types, such as dendritic cells, plasmablasts, etc., we got a few hundred of each of these. Enough to cluster, enough to do analysis. Now, if you downsample that to about 10,000 cells per sample, you'd only get less than 15 cells per sample. And that's not really enough to cluster that out and actually make sense of what's going on there. So by scaling up the experiment to 80,000 cells per sample, you're easily able to pull out these rare cell types. So let's talk about how Parse compares to other technologies, specifically droplet-based technologies. Now, specifically, most of my examples are against 10x droplet-based technologies, but 10x is not the only droplet-based technology available. So with droplet-based technology, there's batch effects, there's logistics. So for example, with these Chromium chips, this same 1 million cell experiment that I just talked about would be run on 7 chips, 7 batches, 50 different lanes on a Chromium controller, and each one of those lanes introduces another batch effect. But with Parse, since we multiplex everything together, that minimizes those batch effects. So this is just an example using mouse brain nuclei. We got tissue from a third-party tissue provider. They sagittally dissected these and flash froze the brains, sent half of each brain to either Parse HQ in Seattle or a 10x genomic certified service provider, ran the experiments respectively, and then compared it. And of course, we see higher gene and transcript sensitivity in Parse data, which means that you'll get more information, more transcriptomic information, and you'll waste less of your sequencing on duplicate reads. Parse data easily combines with any other single-cell data, as you can see here. But where the difference is that we see greater gene expression, which means we have more differentially expressed genes to inform your downstream analyses. Now, this is another comparison, comparing mouse kidney cells. So this was done by another certified service provider, S2 Genomics. They sell dissociation machines. They took these mouse kidneys, dissociated on their S2 singulator, and then performed a red blood cell lysis. Then they immediately split the sample into 2, one aliquot to go into EverCode fixation, one aliquot to run directly on the Chromium controller, and then they sent the fixed cells to Parse HQ to run the barcoding. So with that analysis, we compare these two UMaps, and you can see there's actually a cell type that 10x was able to find with droplet-based technology that Parse was not. This population here, red blood cells. Now, if we look at a marker of red blood cells, hemoglobin, you can see that hemoglobin doesn't show up in any of the Parse data, but it shows up in all of the cell types for the droplet-based technology. Now, one possibility is that all of these cells are transdifferentiating into red blood cells. No, probably not. So let's take a look at this. Let's zoom in a little bit into some of these clusters. So with the droplet-based technology, and for those who have ran droplet-based single cell previously, you're probably familiar with that one cluster right there in the center that shows markers of multiple different cell types. Now, I actually ran into something like this. I was doing developmental studies back in my postdoc. I was doing droplet-based single cell, and I was really excited because I thought I found a multipotent progenitor population that led to both of my cell types. I was super excited. I thought I found something new. I made probes, in situ probes. I made knockouts. I made transgenic animals, the whole 9 yards. And then I showed this to a bioinformatician. And he's like, oh yeah, that's ambient RNA. You want me to clean it up? I'm like, what? I just wasted 6 months of my time on this artifact. And he's like, yeah. I'm like, back to the drawing board. I laugh at it now, and it makes a great story, but it was horrible during my postdoc. So don't waste your time. What you want to do is get cleaner data inherently. With EverCode data, since we use the cell itself as the reaction vessel, you get cleaner data. You avoid that ambiguous cell type identification, and you don't get hemoglobin expressing everywhere, for example. So what ambient RNA is when a cell bursts, whether that's intentionally or unintentionally. When you use droplet-based fluidics, the cell plus the barcoded bead plus any ambient RNA, any RNA that's floating around, get encapsulated in that bubble. So what happens is you lyse this cell, and then all the transcripts that are from the cell, but also all the transcripts that are floating around get the same barcode. So if you have a bunch of cell type specific transcripts just floating around, that's going to lead to some issues. With Evercode technology, with droplet-based, with split pool barcoding technology, we use the cell itself as a reaction vessel. So only what's inside the cell is going to get the barcode. And also, since you need to have a cell and an ambient RNA transcript to travel in the same route for 4 different instances, plus we have some washes built in, the chances that these ambient RNA are going to be barcoded with the same barcodes as your cells is practically 0. So without ambient RNA, we have more well-defined cell subsets, reduced chance for spurious cell type identification, less sequencing wasted on misleading transcripts, and ultimately, and most importantly, less time spent cleaning up your data. Now, I'm going to touch on TCR and BCR just as a very high-level overview. And if you want to learn a little bit more, come find me at the afternoon tea. But speaking of tea, so for the TCR, we're looking at information from the whole transcriptome plus the CDR3 region of the T cell receptor from the same cell. And this is compatible with human and mouse samples. The way it works is you'll do your combinatorial barcoding, you'll get your full-length cDNA, and then you'll split it. One portion to do your whole transcriptome info for short read sequencing, one portion to do your PCR amplification of your CDR3 TCR sequences. And we get high alpha and beta chain pairing efficiency for activated T cells over 85%, and for primary T cells a little short of 75. Now that for those working in T cell biology, that shouldn't be surprising, since the activated T cells are going to be churning out those T cell receptors a lot higher levels than the primary T cells. This is just a example experiment using a TCR mega kit, human T cells from 15 donors, stimulated them with a few samples with autoimmune antigens, a few samples with viral antigens, a few samples with tumor antigens, and a few unstimulated controls, of course. We saw very high specificity. Autoantigen clusters are clearly CD4 positive. Foreign antigen clusters are CD8 positive. And you can see, because of the lower ambient RNA, you don't have a lot of crosstalk or spurious expression of CD4, CD8, et cetera. So you can be more confident that the cells are indeed what they are, and the receptors are truly from the cell types of interest. This is just looking at the EBV paired chains. And by scaling up to larger and larger experiments, you're able to find known T alpha and T beta sequences, but you're also able to find a bunch of previously unknown sequences just by the sheer numbers of cells you're able to profile in one experiment. This is a research spotlight from Nate Redeker-Flynn at Stanford, and I actually personally helped this experiment get off the ground. And he used Evercode TCR to look at mouse metastatic cancers and how the T cells interact with that disease state. And he was able to see very high pairing efficiency. And if you want to learn more about this experiment, there is a webinar available on the Parse Biosciences website. BCR, we capture full-length B-cell receptor sequences. We want full length because of somatic hypermutation. CDR3 is good enough. But for BCR, we want full length, so we'll tile this region, and we get full-length paired heavy and light chains, plus the gene expression from the same cells. This is compatible with human, mouse, and transgenic mouse samples. So the way this works, you'll do the barcoding, you'll do the library preparation, and then you'll do a hybridization pull-down to pull down the BCR sequences. Now, B cells are antibody-producing factories, so we can just do a pull-down instead of doing an amplification. This is just another experiment, million cells, 12 and 12, and we see high heavy light chain pairing efficiency, of course, slightly lower in autoimmune patients, but that's to be expected in autoimmune patients. What's cool about this, though, because we used a mega kit, we see 900,000 plus unique clonotypes because of the ability to scale up to 1 million cells. So that's really the strength of being able to scale up to these larger experiments. Also, at this point, you can start to see the expanded clonotypes because you just have so many possible B-cell receptors. So another great thing is because Parse is a lot more gentle compared to fluidics or droplet-based technology, you can easily profile these rare cell types such as plasmablasts, which are impossible to profile with fluidics and droplet-based single cell. And we can see isotype switching in the B cells as well using Parse technology. So here we see a clear shift of plasmablasts after immunization to the IgG1 isotype. So EverCode TCR, BCR, we see unmatched data quality, high alpha and beta chain detection or heavy light chain detection, TCR, BCR respectively. Means you can detect the true diversity of T cells or B cells in your sample. Flexibility with fixation, this is the only technology that allows paired whole transcriptome plus TCR or BCR information from fixed cells. And of course, it relies and is powered by that exponentially scalable Parse EverCode technology. One great thing about Parse is the support along the way. Jyoti sitting right there is one of the decode field application scientists. So once you purchase your kit, she will be in contact with you to help prepare your experiment, discuss key sample prep and protocol details, and make sure you're setting yourself up for success. We have the Parse Customer Support Suite with video, training, documentation, user guides, etc., to make sure that everything runs smoothly. And we have bioinformatics expertise. Whether you're a bioinformatician or a wet lab biologist, we have tools for you to be able to analyze your data, get biological insights, and make some amazing discoveries. Once again, these are the kit sizes. Mini kit, whole transcriptome kit. Most research I see nowadays, researchers are doing like a WT kit with like 8 to 12 samples, so about 10,000 cells per sample, 10 samples. This is usually where most researchers will start, either there or the mega kit. But once you're ready to scale up to these larger experiments, we have options for that as well. Everything's based on this whole transcriptome information. And we have on top of that TCR, BCR, CRISPR screens, which I didn't talk about today, and gene select for pull downs. And now I'd like to introduce Dr. Osvaldo Contreras for his talk using Parse EverCode technology.